A gene contains genetic information in form of a nucleotide sequence. The nucleotides are adenine, thymine, guanine, and cytosine in DNA, and in RNA, thymine is replaced by uracil. Here we see a strand of DNA. The first step of transcription is initiation. Transcription factors, represented by the green blocks, attach to the DNA and facilitate the attachment of the RNA polymerase, represented by the blue structure. RNA polymerase base pairs RNA nucleotides with the template strand of DNA. Keep in mind that uracil is used rather than thymine. The next step is elongation, when the double helix unwinds as the RNA polymerase adds nucleotides. The last step, termination, occurs when a terminator is encountered. The transcript is then released, and modified if in a eukaryote. Before translation, primary transcripts in eukaryotes undergo processing. Note that the addition of the 5' and poly A tail should precede the removal of introns. The RNA is spliced by the orange spliceosomes, removing introns, or non-coding sequences, leaving only exons. Supposedly preceding splicing, a 5' and poly A tail are added to the 5' and 3' end, respectively. The 5' prevents digestion by enzymes and includes an attachment site for ribosomes. The poly A tail also helps to prevent digestion by enzymes and helps the mRNA exit the nucleus into the cytoplasm. Once in the cytoplasm, the additions help ensure mRNA stability and allows it to bind to ribosomes. Translation is the process by which the codons of an mRNA sequence are changed into an amino acid sequence. The first step is initiation. It begins when mRNA becomes attached to a subunit of the ribosome. The start codon is AUG. It must be positioned correctly in order for translation of an amino acid sequence to begin. The arrival of a large ribosomal subunit completes the initial complex. Amino acids present in the cytoplasm are carried by tRNA, or transfer RNAs, to the codons of the mRNA at the ribosomal complex, according to base-pairing rules. One end of the tRNA bears a specific amino acid, the other end bears 3 nucleotides called anticodons. Each amino acid is joined to the correct tRNA by a specific enzyme called aminoacyl tRNA synthase. Elongation continues as tRNA brings amino acid to the ribosome and a polypeptide chain is formed. The anticodon base pairs with the complementary mRNA codon in the A site. An rRNA molecule of the large subunit catalyzes the formation of peptide bond between the amino group of the now amino acid in the A site and P site. The ribosome translocates the tRNA in the A site to P site. At the same time, the empty tRNA in P site moves to E sites. The last step is termination, when a ribosome reaches a stop codon on mRNA. The A site has ribosome accept a release factor, which promotes hydrolysis of the bond between the tRNA and the P site and last amino acid of the polypeptide, thus forcing the polypeptide from the ribosome. The 2 ribosomal subunits and other components of the assembly dissociate.